Uh, so if you've been here before, welcome back. If you have yet to attend one of these, welcome. What we like to do is send thanks to all of our supporters who make the SDSA activities happen. This includes our inaugural sponsor, the University of Maryland School of Medicine, and of course, generous support from the American Statistical Association. If you have any interest in sponsoring our activities, we have a QR code up here. I will put the link in the chat as well. And we'd also like to thank our additional sponsors. If you've managed to stumble onto this webinar, but you are not a member of the Statistics and Data Science and Aging Interest Group, we would love to have you join us. That way you can keep up to date on some of our activities. I will drop a link in the chat for that as well. And I'd also like to share an opportunity with you that we've learned about. So those of you who are ASA members, you may have seen there's the opportunity to apply for participation in a statistical advisory board for the journal Nature Medicine. They're looking to add some statistical expertise focused on neurology and psychiatry. The caveat here is to be eligible, one must be an ASA member. The idea here is that folks who serve on the board will be responsible for reviewing about 6 to 7 manuscripts annually. The term is 1 year with the possibility of renewal. We have a link here. I will put that in the chat. Please make sure you apply by March 15th. That's when they'll start reviewing applications. If you have additional questions about what this board is all about, Michele Guindani and our ASA president, Ji-Hoon Lee, are the ones who are really spearheading this effort. And I will put their email addresses in the chat as well. Before we get started, I know that Charlie has some announcements as well.
Thanks. I'm Charlie Hall. I'm the chair of the Statistics and Data Science in Aging Interest Group, and I have 2 event announcements. The Biometric Society Eastern North American Region meeting is going to be in New Orleans, March 23rd through 26th, and SDSA will be there. We will have an information table there, and we're also going to have a meetup on Monday night, tentatively scheduled for 8:30, so we can do it after dinner. We're going to walk from the conference hotel to Cafe Du Monde, the quintessential coffee and beignets place in New Orleans that has been in operation since 1864. It's on everybody's list of things must see for tourists in New Orleans. So we're looking forward to that. It's about a 10-minute walk from the main conference hotel. And if you're interested in helping sit at our information table, I'll tell you how to get in touch with me in a moment. And also, we're going to be at the Lifetime Data Science Conference in Brooklyn, which is May 28th through 30th at the Brooklyn Bridge Marriott. And we'll have we're probably going to have a table, and we're going to have a meetup Wednesday night where we're going to walk over either the Brooklyn Bridge or the Manhattan Bridge to Chinatown and have a delicious vegetarian Chinese meal. And if the weather's bad, we'll take the subway. But it'll be an experience to walk over the East River on the Brooklyn Bridge or the Manhattan Bridge. Maybe take one over and then come the other back. I'm going to put a note in the chat. Are people able to do a private message to me in the chat? Question?
Yeah, they can, Charlie.
Okay, so you can either send me an email, send me a text, or do a private message in the chat. Thank you. And you're not here to hear me, you're here to hear Heather. So all yours. Michelle, you can introduce.
Yes, we are pleased to have Heather Chappell, who is an assistant professor at the Wake Forest University School of Medicine, who will be speaking to us about dynamic brain networks and obesity and weight loss in older adults. I will hand it on over to Heather, but I ask for questions. Please put them in the Q&A and not the chat. Thank you so much.
Are my slides in the right view? Or do I need to do something else here?
Good.
Looks good. Okay, perfect. Thank you so much for having me. I'm really excited for this opportunity today to talk about some of my work with you all. So I hope that I could share a little bit about dynamic brain network analyses, and then of course, how we are applying these tools to study obesity and weight loss in older adults. So let's just jump right in here. Inside Out 2 was in theaters this last summer, so I thought it would be kind of fun to use this in today's talk. I wish this was in person so I could poll you all and ask how many of you have seen it. But I'm going to assume that some of you have. I'm also going to assume more of you have seen the original Inside Out movie. But for those of you who haven't seen it, Inside Out is a 2015 Disney Pixar film. One of the film's directors actually conceived of this idea of the film while he was observing changes in his daughter's personality as she was growing older and like a little bit through her teenage years. And he and his co-director actually consulted with many psychologists and neuroscientists in an effort to accurately portray the mind in the movie. So in the first Inside Out movie, an 11-year-old girl, Riley Anderson, moves from Minnesota to San Francisco. And she first thought everything would be great. She was very excited, but then once she moved, she started having doubts about seeing her new house and the other aspects of her new town and her new life. Okay, so that's Riley. And then the other characters in the film are her 5 living emotions. And they are based at the headquarters in her brain. So they have a command center in her brain that they control, which dictates a lot of Riley's behavior. So, for example, Joy is one of the characters, and she attempts to keep Riley happy. So when Joy's at the command center in Riley's brain, Riley's happy, everything's great. You see Riley here, happily running through the living room with her wagon. Fear is another character, and Fear keeps her safe. So here we see Fear take over the controls right before Riley is actually about to run over a cord. She might trip. Fear takes over the controls. Riley pauses right before running over the cord and steps over it very cautiously. Then there is disgust. Disgust keeps her from being poisoned both physically and socially, and sometimes to her detriment, like here when her parents are attempting to feed her broccoli, right? Disgust takes over. She's like, oh no, Riley can't eat this broccoli. And she causes Riley to fling the broccoli at her dad's face. Now, her dad looks happy in this screenshot here, but he's actually not happy. And he tells her that she can't have dessert for dinner because of that. So anger takes over the controls. Now, anger helps her keep her life fair. So when her parents tell her she can't have dessert, anger takes over and causes her to be very visibly upset. And then the final character is Sadness. Now, Sadness doesn't know her purpose at the beginning of the film. But when she takes over the command center in Riley's mind, Riley becomes sad. So you'll have to watch the movie to find out Sadness's purpose. I won't give that away. So where am I going with this? Well, Riley's memories in the film are housed in glass spheres known as memory orbs. And the orbs are colored here by the emotional brain state she was in at the time of the memory. So for example, when she is sad, a blue memory orb travels down a chute and gets stored here in these racks. And that represents the sad brain state she was in. So in a way, when looking at this, we can kind of see her whole brain state sequence. While this is clearly an oversimplification and a cartoon depiction of what might be going on in the brain, there might be some truth to this. So in other words, we may have different repeating brain states that we might transition in and out of. Now, obviously, like in the movie, we do have different emotions and a different emotional states. So yes, we do switch in and out of those. But what I mean is if we actually took snapshots of brain activity over time, should we see some sort of repeating snapshots or patterns? And so my work is centered on this premise that we do have some type of repeating pattern or repeating brain state. And to be honest, I don't actually know if that's true because none of us really know the truth of what's going on in the brain. But I guess I believe that while the brain probably never exactly repeats a state, there are likely repeating patterns. And it would be helpful to us if we could understand some of those patterns and see how they are associated with various brain disorders and diseases. And I believe a network depiction of the brain may actually help us characterize some of those patterns. So before I come back to brain states, I want to briefly discuss what I mean by a brain network. Okay, So before even a brain network, we have to talk about BOLD imaging. BOLD stands for blood oxygen level dependent imaging. And when you get an MRI of your brain, it looks like this donut shaped scanners here. That is the type of imaging that is happening. So basically, big picture, I am not a neuroscientist, but I'm really trying hard to learn these things. BOLD imaging takes advantage of different magnetic properties of oxygenated, so oxyhemoglobin, and deoxygenated, so deoxyhemoglobin blood. And when brain cells are active, they consume more oxygen, leading to an increase in the deoxyhemoglobin levels in the affected area. And these deoxyhemoglobin is more paramagnetic than oxyhemoglobin. So basically, big picture is the giant magnets in the scanner here can pick up on changes in oxygen levels in the blood. And the oxygen levels are kind of a proxy for brain activity. So we can get a measure of brain activity for the length of time that someone is in the scanner across their brain. So I actually volunteered to be a pilot study participant for our studies here at Wake. And this brain up here in the left-hand corner is a picture of my brain. It's a structural picture of my brain. It's just the white and gray matter in my brain. And then this image here on the right is a measure of the functional activity in my brain. So it's actually measuring activity. And if I play this movie here, it's showing the activity changing over time, over a snapshot of time that I was in the MRI machine. Okay, so where do we go from here to construct our networks? Well, we can actually parcellate the brain into many regions at all different levels of granularity, right? So some people do it at the very tiny level and get thousands and thousands of voxels. Or a lot of times what happens in a pre-processing step is a person's brain is kind of warped onto a standard brain atlas or template. And then you can kind of divide the brain into different chunks or areas that are previously known to be like a certain brain region called a certain thing, like the hippocampus, for example. So then we can actually extract the BOLD signal at each of those regions. Whoops, I don't want to play that anymore. At each of those regions, however many you parcellate the brain into, for the length of time that someone is in the scanner. So here we have N brain regions and we have the BOLD activity over time. And those regions will be the nodes in our network. So from there, we actually want to calculate the correlation between every pair of brain regions. Okay. And this will tell us how correlated the brain, the blood oxygen level dependent signals are, which is a proxy for brain activity. The idea is that higher correlation values represent maybe more communication between those brain regions, right? They're more synchronized in their activity. So they're working together in some way. So those are our edges in our network. So here, for example, we get this giant correlation matrix, which represents a weighted network. So every correlation value in this matrix represents a weighted edge between two brain regions, where the correlation value is the actual weight, and that's the strength of the edge. So a larger positive or negative correlation value represents kind of a stronger connection or edge. And to be honest, a lot of the times we pay most attention to the positive connections because we aren't really sure what to do with the negative ones, right? Negative means the two brain regions are anti-correlated, but a lot of times we will focus on the positive ones because we want to see the ones that are positively correlated. So from there, you can plot a network back onto the brain, like in this picture. And sometimes we do a weighted network, just basically putting all of these edges here from the correlation matrix onto the brain, and then representing the weight of the edge by the thickness of the edge here in the plot. And then a lot of times, though, what people will do is they'll binarize the network. They'll say anything above a certain correlation value, that means there's an edge between those regions, and anything that's not is a non-edge. So for the longest time, I would say for like the first 10-ish years of where brain networks were really a thing, which probably started around like the 2010-ish timeframe, people did static connectivity, where they took the time series for the whole length of time someone was in the scanner, and they got one giant network, so one average network. But in the past 5 or so years, what people have been really interested in is dynamic connectivity, because it makes sense that you would be transitioning in and out of multiple brain networks while you're even at rest doing nothing because you're thinking about all different things, or while you're performing a task in the scanner, you might be switching of different networks. So now dynamic connectivity has really come to the forefront of network brain science. And really, why networks to begin with? Why did networks become important? Because for the longest time, we actually attempted to pinpoint regions that controlled specific functions. So for example, what part of the brain is responsible for memory, or what part of the brain is responsible for motor movements? And that was helpful, but an oversimplification, right? Because there are no areas solely responsible for complex functions like language, emotion, attention. Multiple areas are working in complex coordination, right? And the manner in which all these different areas are connected is highly informative. Okay, so now let's return to the idea of brain states. So ultimately what I want to do is estimate what someone's brain state sequence is for the length of time they are in an fMRI machine. And instead of characterizing these states like by emotions like an inside out, instead we're characterizing them mainly by a brain network. So each state will represent a different weighted brain network. And this is a state-based dynamic connectivity analysis. So here's an example that I'm going to come back to later and show you results from of a weight loss study in older adults that happened here at Wake Forest University. And it looked at the efficacy of 3 different treatments on weight loss in a group of older adults. What I became interested in was whether their baseline fMRI scan, while they were performing a food cue task, so they were shown images of their favorite foods, could tell us something about what is going on in the brain that would be associated with whether they were successfully able to lose weight after 6 months on the intervention that they were assigned to. So is there just something baseline about what is going on in their brain that would be associated with whether they can successfully go on to lose weight? So in this example here, right, let's say we fit 5 brain networks to the data, and we see that this individual starts in this blue state, this blue network state, then sees a slice of pizza, but then switches to this green network state, whatever that is, then goes back to blue, but then sees an ice cream cone and is into a completely different state, right? And so we're interested in what those states actually are, what the brain networks look like, but also the dynamics here, right? So for example, the transition probabilities. What's the probability they go from blue to green versus from blue to orange? Or their dwell time distributions. When they enter a specific state, like the blue state, what's the probability they stay in that for a long amount of time versus the probability that they switch out of it pretty quickly? These are all questions that we want to answer and then compare across different groups of people, like in this case, the people who successfully lost the weight versus those who didn't. And then this is just another depiction of what we're trying to do here, where we have the bold signal at each brain region, and we want to be able to tell what states this person is in and when they're transitioning to different states. Okay, so how can we do this? Well, what people most often do for dynamic connectivity analysis is what is called a sliding window analysis. And people do this because it's simple. It really doesn't take much coding. It's built into a lot of the standard fMRI analysis toolboxes. So it's basically just like it sounds. You take all the time series data, at each region for the length of time that someone's in the scanner, and you choose a window size or length, and you take the first chunk of the time series and you calculate your correlation matrix or your network, and then you shift the window and you do this again, and you shift it and you do it again, and so forth. And then from there, if people actually want to group these into different repeating states, they can do some sort of clustering analysis, like a k-means clustering analysis. The issue with this is that it has been shown that people are unable to capture quick changes in dynamic connectivity with this, right? Because you're kind of averaging across a window. And so if someone switches, from one time point and they're in one state and then they go immediately to another state and then right back or something, you might miss that with this type of analysis. Also, what do you set your window length to be, right? If you go too long, then you risk not being able to capture these quick changes. If you make your window length shorter, then you might not have enough data, and then your estimates are not stable. So there are issues with this approach, but people still like to use it because, as I said, it's simple. So then what people have started to use a little bit more now are hidden Markov models. And a major pro to this method is you don't have to set that window length. So just a quick overview here. You have your observed data here, that's your Ys, right? That's the observed bold signal at each of the regions that you've parcellated your brain into. And you have this at each time point in the scanner. We actually call that a TR, the repetition time. And technically the TR is the amount of time that passes between consecutive acquired brain volumes, but we just say sort of the first snapshot of brain activity happened at TR1, and then we have our second TR and third TR and fourth TR. So when you hear me say TR, that's what I mean by that. So what the hidden Markov model is saying is that we have our observed bold signals. That's what we observe, but there's a hidden layer underneath, and those hidden states are what we are most interested in, and they give rise to the bold signal that we are actually observing. And the hidden states represent unique brain networks of distinct activity and functional connectivity that repeat at different points in time. Okay, so what we say is that the Ys, the observed bold signals, follow a multivariate Gaussian distribution dependent on the state that someone is in that we don't know, but we want to estimate. So the mean and covariance matrix are dependent on that state. And the covariance matrix can be converted to a correlation matrix, which is basically our network. So every state has its own network. And so we can write out the log likelihood, and you see we have the first part, which is our Y given S, right? That's the multivariate Gaussian distribution. The next part here, we have our transition probabilities from our Markov chain. So probability of going from blue state to red state, from red staying in red, and so forth. And then this last term here is the probability of just starting in a particular state. Now, the one limitation of this is that implicit in this model assumption, right, just based on the fact that your transitions between states just kind of follow a basic probability transition matrix. The probability of remaining in a state for a certain number of TRs actually follows a geometric distribution, right? Because the diagonals of your transition probability matrix, you can think, okay, the probability of staying in state 3 for 4 time points and switching, you would pull out the diagonal associated with staying in state 3, right? Times, that times that times that times that times 1 minus that. That's a geometric distribution. And with the geometric distribution, it's increasing like this. So what that is saying is you have a high probability for only spending a couple of TRs in a state before you jump out to another state. And what if that is an unrealistic assumption, right? And so it actually is encouraging faster state switches, which might not be true. So in my work, I have proposed using a hidden semi-Markov model instead. And I'm not going to go too much into the math here because I want to get to some of our results. But the main difference with the hidden semi-Markov model is that we now have the sojourn distribution, that's the DUR here, explicitly in the model that we can estimate. And it doesn't have to be geometric. You can define that to be a Poisson distribution or a gamma distribution, or if you have no idea what that sojourn or dwell time distribution is, it could be a non-parametric distribution. So we're not assuming a geometric distribution. And all of our other model parameters essentially stay the same. We have the multivariate Gaussian distribution for the Ys given the S's with the mean and covariance matrix for each state. We have our transition probabilities, but now those transition probabilities are when you leave a state, where do you go next? So they're conditional transition probabilities. And then you still have the probability of starting in a particular state. So the main change here is the addition of the sojourner dwell time distribution that is going to capture your probability of remaining in a state for a certain amount of time before you switch out of it. So these model parameters are estimated via an EM algorithm, an expectation maximization algorithm, to get the maximum likelihood estimates. Now, we can fit this model to a single participant if we want. So if somebody has a particularly long scan time, you actually can fit it just on one participant. A lot of times what we do is we fit it to a group of participants. And we do this so that we have more data, because there are actually a lot of parameters to estimate here, right? For every network, you're estimating that correlation value between every pair. You're also estimating the dwell time or the sojourn time distributions. I use dwell and sojourn interchangeably. The transition probability, and also the probabilities of starting in a given state. So there's a lot to estimate. And if you want to have some larger brain networks here with hundreds of regions, right, that's a lot of parameters. So typically we'll fit it on a whole group of people. And from there, we will get a set of brain network states that the individuals are transitioning in and out of. And then what we're really interested in is, how do the dynamics change, right? Do some of those people in the group spend more time in state 1, and maybe some others spend more time in state 5? Maybe one group dwells longer in state 1 and the other jumps out of it more quickly. So we get our group of states that we fit from the whole population. But then we're interested in making those comparisons. So we'll split into our 2 different groups. So let's say younger versus older, for example. And we will rerun the model on each group separately, fixing the states, but getting group estimates of the dynamics, like the dwell time distributions and the transition probabilities, because then we can make those comparisons. We could, fit the model on our 2 or 3 or however many groups we're interested in separately. Then you're going to get different sets of brain states for each one. And then how do you kind of compare the dynamics across groups, right? Because state 1 for one group will be different than state 1 in another group. But that is something that can be done. Okay, so now let's get to our application to a study on aging and obesity. So first of all, why is this important? Well, we all know US obesity prevalence is rising, right? And severe obesity increased from 4.7% to 9.2% just from 2000 to 2020. And I should probably get some updated stats there. But obesity-related conditions include heart disease, stroke, type 2 diabetes, certain types of cancer, and older adults are already vulnerable to those chronic diseases. So it's extra. They are extra at risk than if they also have obesity, right, to be suffering from some of those. So there's an urgent need to understand the struggle that many older adults face with intentional weight loss and maintenance. So participants for the analyses I'm going to talk about involved a sample of older adults with obesity that were recruited from the Empower study. And the Empower study was a study that happened here at Wake Forest University. The purpose of it was to determine the extent to which 3 different treatment conditions promoted short-term weight loss and longer-term weight maintenance. So there was a sample of 71 older adults. That's what I'm working with, that were kind of recruited for this ancillary study that involved MRI or fMRI analyses on their brains. So all participants completed an in-person screening visit where they were asked about their favorite foods, and then a 45-minute MRI scan after an overnight fast of at least 8 hours. And here were some of the inclusionary criterion. So ages 65 to 85, a BMI between 35 and 45, minimal activity levels, weight stability, so not really much change in their weight over the last 6 months. And obviously, they had to be able to do safe and optimal participation in exercise training. So the different treatment conditions were One group had a dietary weight loss program with a structured aerobic exercise throughout the week. The other had a dietary weight loss program with a daily movement program. So a certain number of steps, certain number of times standing up. It was called the Sit Less program. And then a third group that had all three of these, the dietary weight loss program, the structured aerobic, and the Sit Less program. So as I mentioned, they shared their 4 favorite foods. And then during their MRI scan, they had a food cue visualization task part where they were shown words of their favorite foods. So that was kind of interesting to me when I first heard about how they were shown words instead of actual pictures. As I showed you earlier, I had actual pictures of their favorite foods. The reason for words is because if I showed you a cheeseburger, maybe, and I showed it to you with pickles and ketchup on it, that might not be appealing to you, even if cheeseburgers are your favorite food, right? Because you might prefer your cheeseburger without ketchup and pickles. So the word was presented, and the participants were told to imagine the taste and the smell and the satisfaction of consuming that food. And what this was getting at, these food cues, was the hedonic drive to consume food. And what that means is your drive to consume food, even if you're not necessarily hungry, just consuming food for pleasure. And this has been shown to be associated with weight loss success and weight loss maintenance. And it has been shown to elicit broad effects across the brain. So one way that this could be measured is via the power of food scale. So I wanted to show you some examples of some questions here that these food cue tasks are trying to get at to tap into that hedonic drive, right? And so, for example, I find myself thinking about food even when I'm not physically hungry. I mean, that's definitely me. When I taste a favorite food, I feel intense pleasure. If I see or smell a food I like, I get a powerful urge to have some. Just before I eat a favorite food, I feel intense anticipation, right? So higher scores on this would mean that like food has a lot of power over you. And it makes sense that this type of drive, this hedonic drive to consume food, will be associated with your ability to lose weight. And it has been shown that those food cue tasks can tap into this. So I have not yet looked into any of how well the interventions are associated, specific ones were associated with the brain. What my main question so far has been, are there signature brain network states and patterns of state traversal that are associated with successful weight loss in older adults with obesity, no matter what intervention that they are on, right? Just, is there something at baseline before they even do any type of intervention that then goes on to be associated with how well they do after the intervention? Do they successfully lose weight? So for our analyses, we looked at 58 participants. We had to remove some of the participants from the analysis because of some data that wasn't great. A lot of movement in the scanner, for example, would mean that we had to cut some people out because we weren't getting a great signal. So we focused our analysis on 58 regions that were previously found to be associated with weight loss in an independent data set, not with this type of analysis at all. It was actually a machine learning based analysis, and they came out as regions that were potentially important for weight loss. So we focused on those. We used the 58 regions on the 58 individuals, and we defined weight loss success after 6 months as losing at least 5% of your body weight. And then we fit the hidden semi-Markov model on the data. All right, so this isn't telling you a whole lot here, but I wanted to at least show the picture because we got our model estimates, and from there you can actually use what's called the Viterbi algorithm. It's a dynamic programming algorithm to estimate for each individual their most likely state sequence. So here, the rows are for the participants. So every row is a different participants. And you'll see, like, for example, the first participant started in this green state transitioned to this other state, which I'm coloring yellow, went to red, and then went back to green, and so forth. So we have this state sequence for each person. Okay, so the first question we typically like to try to answer is, okay, well, how much time are people spending in each of the states? So we looked at this for the unsuccessful weight loss group and the successful weight loss group. And what we saw was that states 1 and 2 in particular spent different amounts of, or sorry, states 1 and 2 in particular had different amounts of time spent in them for the unsuccessful and successful groups, right? So for example, the successful group spent more time in state 1, the p-value is 0.025, and less time in state 2, p-value 0.031. And then states 3 and 4 were kind of borderline significant, where the successful group spent more time in state 3 and less time in state 4. And then state 5 was actually kind of this dominant state that both groups spent a large amount of time in. The difference was not significant there. And I'm going to get back to what the states actually are, but I first wanted to show you transition probabilities, because this is really interesting too. So I have a figure here for successful and unsuccessful. The size of each of the circles here represents or is proportional to the amount of time that was spent in that state. So these are each of the 5 states, right? You can see both groups spent a lot of time in state 5. So I made that circle larger. The arrows represent the probability of transitioning from one state to another. Thicker the arrow, higher probability of going from that state to the other state. So for example, if we look at state 2, since that was one of our main players here, that had a significant difference in the total time people were spending in the states, right, between the successful and unsuccessful groups. You might be wondering, it's interesting because there are smaller arrows going into state 2 for both of these groups. So it's kind of seeming like, people aren't really transitioning into that state much. What does that mean? Especially because the unsuccessful group actually spent quite a bit of time in state 2. Well, if from there you then look at the dwell time distributions, which tells you your probability of remaining in a state when you enter it. So if we look at state 2 for the unsuccessful group, that's the blue line for the unsuccessful group. Whoops, keep accidentally touching my screen and then it shifts to the next slide. So if you look at the blue line for the unsuccessful group, it's a little bit flatter. And for the successful group, it's has a peak more at the beginning, right? So what's that saying is the successful group has a higher probability of jumping out of it pretty quickly compared to the unsuccessful group that has that flatter curve, right? They have some probability, some substantial probability of remaining in the state for a while, not just 1 or 2 or 3 TRs. So it's really important when trying to tell the story that you look at the transition probabilities kind of in combination with these dwell time distributions. So I'll go back now to the transition probabilities. Something else to note is the unsuccessful groups seem to cycle between state 3 and 5, right? You see like thick arrows there, which is kind of interesting because state 3 was a little more prominent in the successful group. But if you go to the sojourn or the dwell time distributions, it shows that they jump out of that group pretty quickly. Jump out of that state pretty quickly. And for these dwell time distributions, states 1, 2, and 4 were significantly different in the dwell time, or I should say the successful and unsuccessful groups had significantly different dwell time distributions for states 1, 2, and 4, all with p-values below 0.05. Okay, so now let's get to what are the actual brain states, because it seems like states 1 and 2 were really key players, right? If we go back to the occupancy times, those were the most significant. State 1, where the successful group was in more, state 2, where the unsuccessful group was in more. Now I could show you those correlation matrices for each of the brain states, but sometimes they're kind of hard to read just because, you have like 58 by 58, and it's kind of hard to get a good picture of what's going on in the brain, especially because they're not mapped kind of back onto the brain when you're looking at it in that view. So what we actually did here is we did this modularity analysis for each of the states, and it's like a community detection based analysis. And what that is doing is saying, let's figure out the modules, meaning the communities within the network. So regions within the same modular community are more highly connected to each other and less connected to regions in other communities. So you'll see here different colors. Each color is a different module. So anything in green, for example, in state 1 is highly connected to each other and less connected to regions in the other colors. And then we did that for each state separately. So if we look at state 1, which is where the successful group spent a lot of time, okay, what I want to draw your attention to is that the insula here and the limbic regions are all red colored, meaning that they're in the same module and they're highly connected. And then the motor regions are a different color. So they are segregated from these regions. They have few connections, but the motor has few connections to the limbic and the insula regions. And then interestingly, in state 2, the motor and the insula are in the same module. They're green colored here. They're highly connected, and the limbic is off on its own. And so talking with some of my neuroscience collaborators, and thinking about, does this make sense? First of all, the insula and limbic regions apparently show up a lot in the weight loss literature, but they thought this was particularly interesting because the insula is important for tasting, risk-reward behavior, regulation of bodily function. It's because of the insula that people have the awareness about their body and self. It kind of controls their appetite and the regulation of energy balance. It tells you, you need food, you should eat. The motor actually drives you to eat. That actually allows you to pick up your fork and eat, or, use your hands to eat the cheeseburger. And the limbic processes emotions. It kind of mediates motivational reward aspects of feeding. And that might be activated when you're kind of thinking a little bit about, how do I feel? Am I really hungry? So the thought was that maybe, the unsuccessful group spent more time in state 2, where their insula and motor are highly connected. So perhaps, they see the picture or the word of their favorite food and they think, oh, food, like I might be hungry. And then immediately they imagine eating it, which is why the motor is activated at the same time. Whereas the other group that is able to successfully go on and lose weight spent more time in state 1, where they don't immediately have that connection to the motor region, right? Instead, they sort of have this connection to their limbic regions, and maybe they think about it for a while, like, am I actually hungry? Am I motivated to eat right now? They give it some thought instead of immediately visualizing eating. So again, we don't really know, but it was interesting that we saw this stark difference. And we did have this significant result of these two groups spending different amounts of times in these two states, where these two states really are different with respect to their connections between the motor, the insula, and the limbic regions. So the brain is a complex dynamic system, and the hidden semi-Markov model can be to identify brain network dynamics. We saw that successful weight loss in older adults is associated with the insula being more strongly associated with emotional regions rather than motor regions, which was interesting. It's too early to have direct clinical impact, but the hope is that we can identify signatures of brain or signatures of weight loss success in the brain that can be used to maybe personalize interventions someday, right? Especially in the older adult population, where they're especially at risk for some of those risk factors. So maybe, or for some of those conditions such as obesity, or not obesity, sorry, some of those conditions such as diabetes and heart disease. So maybe, for example, someone has brain organization that is amenable to behavioral interventions alone, and you could avoid using medications that likely, would require lifetime administration. We would really love to do this type of analysis on a younger group of participants as well, to see if we see similar patterns as to what we see with the older adult population, or if this is something that is very unique to just the older adult population. We think that that would be very interesting. Don't have the data for that currently, but that is something that I think we would love to do in the future. Also, there are several statistical challenges. So, for example, one of our biggest challenges is, how do we choose the number of states? So just to talk a little bit more about that, what we have found through simulations is that when we fit the hidden semi-Markov model with too few states, because you actually need to specify the number of states before you fit the model. Like, I have to say I want to fit 5 or I want to fit 6, right? If we fit it with too few, when we know the actual true number through simulations, the states resemble mixtures of the ground truth states, and as a result, they tend to be more similar to each other. And then if we fit too many, states also become more similar, because one kind of true state tends to be split into highly similar states with some attributes of other states. So again, we're seeing more similarity between the states. So it seems like the optimal number of brain states tends to be the number which kind of allows the states to be the most distinct from one another. And what we really need then is a better way to determine how distinct the brain states are from each other. So some sort of distance measure that can measure the distance between the network states. And there's actually, one of my students right now is working on this problem of coming up with such a distance measure. And what we found is that it's really important that these distance measures can calculate information at different levels: a low level, a mesoscale level, and a high level. So low level scale meaning information at the level of the individual nodes and edges within the network or the graph. So for example, something like Euclidean distance. But when you look at that in isolation, you kind of miss insights gained from looking at interactions within the network. So then incorporating something from the mesoscale level that will look at some of the network topological properties, such as global efficiency or shortest path length, that really gets at the ability for the system to efficiently transfer information throughout. And then finally, information embedded in the network at a more global level. People use spectral methods to do things like this, where it's based on the Laplacian matrix of the adjacency matrix, right? And this would be a great way to get some of that global information. So he is basically working on a measure that will incorporate all 3 of those levels into one distance measure. And we're hoping that that will allow us to better be able to determine what the correct number of states is to fit to a given data set. Some other statistical challenges. Right now, we can't really count for covariates in the model, right? We can't really adjust for things. We can do that with some post hoc analyses, but nothing that is directly built into the model. Another issue is the fact that we have to typically work with smaller network sizes because of the number of parameters we have to estimate. So being able to work with larger network sizes, so we don't have to have such large regions, right? We can parcelate the brain into smaller regions and kind of have less averaging going on of the signal, would be very helpful. And we've started to work on a penalized version of this model that will shrink some of those network covariance estimates to 0 and allow for us to work with larger networks. And then probably I would say maybe the most important thing for right now is how to make this analysis easier for researchers to use, because neuroscientists are using the sliding window approach because that is what is easiest. So if we could get this out there in a way that allows researchers to kind of plug their data into a toolbox and use it, that would be amazing. And the student that I mentioned, Mohammed Reza Kodai, who's working on the distance measure, has actually built a toolbox that will allow for this. Now it's still in the beginning stages of testing, and the paper is currently under review, but we hope that that will be available soon so that more researchers can use our methods. And lastly, I have my acknowledgement slide here. This has definitely been a team effort here. I am part of the Laboratory for Complex Brain Networks here at Wake Forest University, but also collaborate with some faculty in the exercise science department and some faculty at Johns Hopkins University, where I did my postdoc, where a bunch of this work actually began. And then obviously the funding sources wouldn't have been possible without funding. So thank you for having me, and I'm happy to take some questions now if there's time.
Heather, that was an amazing talk. Thank you. We did have one question in the chat that you largely addressed, which is getting at the how we defined the number of states, and it got me thinking. This is from Shan Yao, but her question got me thinking about how in a lot of latent variable methods, what we would typically do is fit our model, with multiple number of categories of whatever latent variable, and then use some sort of measure of goodness of fit, and then look for where you have kind of diminishing marginal returns. Is that somewhat what's done typically in this example as well?
So actually, when I first started with the model, I did try to use a cross-validated BIC criterion, but it took a while to run because each run of the algorithm, takes a little bit of time. And then to be doing it so many different times, it got to be too computationally expensive, basically. So then we started to do simulations, and we played around with, what happens if we fit too few, too many? Is there a way for us to tell when we're fitting too few and too many? And then that's when we noticed the distance stuff and how states ended up looking very, very similar when we were fitting the wrong number of states. And it's hard because in the brain, like, do we actually have a true number of states? It's hard because it's even with the simulations, I don't know if that directly translates to real data. The other thing I will say is oftentimes we are limited with the number of states we can fix just based on the sample size we have. We can't go and fit 25 states because we just don't have enough data to do that. So a lot of times I think what we're doing is fitting kind of the most we can for the amount of data that we have. And we'll run the EM algorithm multiple times with different starting values. And we make sure that we're getting very similar results from each of those runs. And if we're not, or even if we're getting different results, with the same starting values each time, that's a way for us to know that maybe we're trying to ask too much of this data and we're fitting too many states, and then we kind of back off. So honestly, a lot of the time we're restricted mostly by just, we can't do more because of the sample size.
Got it. Okay, there's another question, which is, this is also from Shan. So she's asking, can the algorithm be used to group people and link the groups to other characteristics or health behaviors? So for example, can we investigate whether those who stay a lot of time in state one were disproportionately more likely to take specific medication?
Oh yeah, that would definitely be something to look at. And that kind of gets to at the extension that we want to do, which we've started to work on a little bit of, like a building covariates right into the modeling framework, right? So that we can actually directly test, whether medications or I don't know, age or sex or any of those things are directly dependent on the dwell time distributions or the transition probabilities, instead of having to kind of, fit the model on everyone and then divide into groups and then do like an ad hoc thing, like I mentioned, where we fix the states and, rerun the model on each group separately. It would just be amazing if we could just put variables right in the model and directly test that way.
Yeah, exactly. I was thinking the same thing. If you could just, for example, since this was
Yeah.
in the context of an intervention study, if you can put intervention group, as your covariate right
Right,
in there.
right.
We have another question. This one is from Wenyu Gao, who is saying, thank you for an amazing talk. And for the transition matrix, do you assume that all probabilities remain the same over time, or is it possible to consider that it changes dynamically?
Yeah, so right now we do assume that it's the same over time, but that's a great point. I think it's very realistic to probably say that's not actually an assumption that always holds. And so, if we could develop a model in the future that would allow for the transition probabilities to change, that would be amazing. Again, that would require more of the data, more parameters to estimate, but definitely something that would be great to do for the future.
Great. I have a question, and this is something that I may have missed when you were speaking, but in terms of the semi Markov model aspect of this, you were saying that you basically relax the assumption about the sojourn time distribution.
Right.
Is this something that's left non-parametric, or would you have to specify a
Yeah.
parametric distribution ahead of time?
So you can do either. I usually leave it as non-parametric because I'm not sure what the distribution should be, but sometimes what I've done is use non-parametric, kind of get a rough estimate of it, and then say, oh, it looks kind of Poisson, or maybe I can fit a gamma to this, and then I go back and rerun it and actually define a parametric distribution. But usually, most often than not, I do non-parametric.
Great, thank you. All right, does anyone have any other questions? I see, according to my clock, it's 1:58. Ah, we have a taker. Great. Okay. One last question is, thank you for the great presentation. I wonder how this lab work can be transformed into the development of clinical interventions for older patients. Maybe this has been talked about already. I may have missed it. Thank you.
Yeah, that's a great question. I mean, obviously that would be the ultimate goal, right? We're still pretty far from that at this point, I think. I think first of all, these results would need to be replicated in other studies, which we hope to do. But then it's not immediately cleared. Like, just because we know, which brain regions are important, I don't know yet how those specific areas could be targeted. I mean, some people do have targeted therapy done on different brain regions, like TMS or some other treatments, but I'm not sure yet. Maybe medication, right? If we find that certain medications can control certain brain regions better, but I think now we're just trying to figure out, what parts of the brain are important, and then from there we can start to think about treatments. But we're a long way from that, unfortunately.
Hey, getting in on the ground floor then.
Yeah.
Okay, great. So thank you so much, Heather, for presenting today. I'd also like to thank Donna Lalonde at ASA for helping us to set up these webinars, who've been an amazing support. Thanks as always to our supporters and collaborators and our members. And we are going to be having a webinar next month. It's going to be James O'Malley. The topic and title are TBD. I'll reach out to all of you. If you have colleagues who you think would be interested in this interest group, please send along the information. We'd love to have everyone. So thank you and enjoy the rest of your day. Bye, everyone.
Bye. Thank you.